I hope by the end of my talk you will realize that fungi is not only good for wine, some cheese, but also it's going to help you for your health as well. So for a long time, I've been talking about, not just the bacteria, and now, of course, we heard about the viruses are the only microbiome. Really, we have also the fungi. And I want to start to telling you why I'm surprised, in spite of the fact that more than 43 years ago, I figured out that fungi and bacteria live together. So my master degree supervisor in England, he gave me a paper and he said, this is your project. And it was a rabbit treated with antibiotics, and guess what happened? They developed fungal infection. So I figured out that, these bacteria and fungi, they live together, and they really, if you don't disturb one, or if one of them is disturbed, the other will take over. And again, that was 43 years ago. Now in 2010, I tried to also talk about, I don't think this is working very well, or maybe this one will. Oh. Yeah, I tried to talk in the Microbe, which is the ASM, sort of newsletter, saying, look, it's time for us to start looking at not only bacteria, but also fungi. And because I believed at that time that if you study the two together, you are going to have a better understanding of how microorganisms can affect you. But to no avail, nobody listened. But I'm not a shy person, so I continued to do again. In February of 2016, I wrote an article in The Scientist, and I called it The Ignored Kingdom, where the microbiome or the fungal community, nobody wants to listen. But the good news is we are starting to see more of that. In that article, I showed that we did search for what are the organisms that or what are the studies that look at the microbiome. And you can see there are more than 6,000 articles where, I don't know what happened, it changed around this. I don't know, I'm not touching anything, just for you to know. So more than 6,000 articles had microbiome, and only 270 articles ever mentioned fungi. And if you look at 2015, you see 740 articles where you talk about bacteria, 12 only about fungi, and 31 about the virome. Now, we are starting to see a little bit of the change. And here you can see that a number of cavities in the body, they looked at the microbiome, and as you can see, we find the fungi everywhere. And the ones in red here are those are known to be pathogenic. Okay, so now I want to tell you about the story where we start looking at the fungal element, where we looked at oral cavity of healthy people and tried to look at what is there as far as the fungi is concerned. This is the study design, 20 healthy subjects, and you can see the demographic, no need for me to go over it. What's really important is that we did oral wash, not just saliva or a swab, but we took really the total what is in there. And as you know, with the fungi, we use the ITS region because this is designed to detect the consensus sequences present in a wide range of fungi, and this is what we do these days. So in that paper, we showed that there are 74 culturable and 11 non-culturable genera, fungal genera. There's a huge number of organisms. And what I liked is that the number of species identified were 101. It's like 101 Dalmatians, okay? So you can see these are the different organisms. But what we did also, because of the idea of contamination, or like you eat some nachos and then suddenly you have the fungi in nachos, we looked at what are the fungi that present in at least 20% of the population or we studied, and that represent the core microbiome. And these are the organisms which we saw. To no surprise, Candida is by far the most abundant, a lot of non-culturable, and then Cladosporium, you see Aspergillus, Fusarium, and Cryptococcus, which, as you know, these could be pathogenic, especially in HIV, where, these organisms could be there, like Cryptococcus, and you have cryptococcal meningitis there. So our conclusion is that the humans are colonized with numerous fungi. It is really a lot of variation like you see in bacteria between individual to individual. And really at the end, we suggested that it's very good to look at both bacteria and fungi in the same samples. And now I'm going to tell you about the microbiome in patients. And I'm going to go into two diseases. One of them is the microbiome and HIV. Here, again, we looked at the oral cavity, and we characterized what is the microorganism in HIV-infected and uninfected patients. Again, we used oral rinse. We looked at the uninfected controls were recruited from the staff students at Case, and the HIV patients were from the ACTU at Case Western. Now, this is when I'm showing you bacteria, because when we go and try to publish on the fungi, people say, look, bacteria is more important. We say, okay, let's satisfy them. Let's look at bacteria. And here we showed about the oral Bacterium, and this is uninfected and HIV infected. This is a little bit more looking at really what is elevated in the blue in the uninfected and in the red in HIV infected. And you can see there is a number of organisms like Haemophilus, for example, in the blue, and you have Veronella, Neisseria, Porphyromonas as well is increased in HIV-infected patients. So in total, 9 to 14 taxa were present in the HIV-infected, the similar type of thing, and this is what the organisms we saw, or the genus we saw in these patients. This is the core oral microbiome, Bacterium, and you can see this is uninfected. Most of the organisms were common to the two groups. Only Agrobacter was in the infected, and Capnocytophagia was in the HIV infected. This is the picture now with the microbiome, again, uninfected versus HIV infected. And this is, again, just to go to the point, again, elevated in HIV in red. And you can see Candida, it's really huge number of organisms present. It's increased in HIV, whereas the other organism which I want you to watch is this Pichia. Pichia, or sometimes you call it Pichia, and it is also a yeast, okay? So with that in mind, here is again, the core microbiome. You have, these are common, Candida and Penicillium to the two groups, and you see in the uninfected Pichia, Cladosporium and Fusarium, whereas the HIV-infected Alternera, Epicoccum, and Trichosporum. So we noticed that there is a shift in the microbiome between the two groups. We also looked at correlation. I think this is one point which is really very important, which I did not see much in the talks here, is what is the correlation between bacteria and fungi in both population. And I think this is, you will find, as you will see, is very, very helpful. In the infected, we found 15 bacteria-fungal pairs where they were significantly correlated, and in the HIV were 12 of them. Most of them, 11 of the 12 were positive correlation, and only one negative correlation was with Campylobacter. So with that in mind, we really looked at the oral health, healthy people, then in HIV, not HIV, and we published a number of papers on those. The 2010 where we talked about in healthy people, and then we talked about the microbiome in HIV, and the last one we published a paper where we looked at the metabolome as well of these patients. One thing which because I'm a Candida person, I tried to look, is there a correlation between any of these organisms? And lo and behold, when we looked at the people, we found that people who have Pichia, which is dark, they tended not to have Candida, and the other way around in general. So we hypothesized that it is possible that Pichia antagonizes Candida. So to test this hypothesis, we have a huge collection of fungal strains in our center. So we got some Pichia as well as the Candida and tried to see whether they affect the growth, the biofilm formation, and various virulence factors. And this is some of the data where when you look at effect of Pichia spent medium, and this is Candida, Aspergillus, and Fusarium. This is untreated with a Pichia supernatant, very nice growth, and you can see a really significant inhibition when you put the Pichia. So our studies showed that PSM, which is the Pichia supernatant, inhibited Candida, Aspergillus, and Fusarium. The other thing which it did is if you have untreated Candida, as you know, one of the major features of or pathogeneity factor for Candida is ability to form hyphae. So very nice hyphae here. I know you can't see it much. It's very clear in here. But we saw stunted hyphae. In other words, it affected the germination process of the Candida. We also looked at activity of Pichia in the biofilms. And here you can see Candida grows very significant biofilms. And when you treat it with Pichia cells, as well as supernated at different ratios, we had significant reduction in the biofilm ability of these organisms, of the Candida growth. Now, this is some confocal microscopy where it shows you untreated, penicillium treated, because penicillium was present in equal proportion if you in both healthy and HIV-infected patients. And you can see a little, not really much effect. When you treat it with Pichia, the biofilm basically disappeared. And when you look at the thickness of the biofilm, which is here, you can see untreated, Pichia treated, and then with penicillium, no effect as well. So it's clear that it affects not only growth, germination, but also biofilm formation. So this all nice and dandy in vitro. What we did then, we developed a oral candidiasis model, mouse model, and we treated with Pichia supernatant or topical nystatin, which is the standard of care for oral candidiasis, and we had untreated and vehicle-treated control. We did the treatment for 7 days, and then we had a scoring as well as histology for how well they did. And this is here the initial infection score. You can see here vehicle and untreated, nystatin, and then the PSM, and there is a significant reduction in that. And when you look at CFU, again, treatment with the Pichia supernatant really reduced it. And a picture worth 1,000 words. Look at this untreated, nystatin treated, and very, very few hyphae or stunted, really, hyphae in PSM treated, which tells you that this is a really a potential approach to treat this organism. So now we are working on the proteins. I'm not going to tell you too much. We are still working, trying to identify what sort of the components inside that supernatant, okay? And we believe it's a proteinaceous in nature. This is some of the data. And when you try to treat with protein inhibitor, pepstatin, for example, you have an increase back in the biofilm formation there. So now, recent work with Michael Lederman and Nick Vanderberg, we're starting to look at, he has a very nice cohort, a glyph cohort, healthy individual. They were treated with antiretroviral therapy, but they in failure, basically immune failure and immune success. And we're trying to look at both the microbiome and the bacterium, and you can see that in the healthy is very similar to the immune success, whereas in immune failure, the diversity is quite low. The same with respect to the bacteria. We are also looking at what is the bacterial abundance, and here you can see that there are some differences, again, based on the colors, and this organism, particularly Colencella, it's really upregulated in the IS as well as in the immune failure. Now, this one thing we looked at, this organism, Colencella, seemed to have like it promotes sort of obesity. We did not have all the data about obesity in these patients, but we were able to look at the BMI And doing regression model analysis, we found that linear regression model analysis, BMI of IF patients were associated with abundance of Colinella, and really Colinella have been reported in the literature to have a risk factor for heart attack and also linked to alteration in the serum lipids. So this is what we are gonna do from now on, trying to tease this apart in a collaborative work. What about the microbiome and Crohn's disease? So I want to change a little bit to show you some of our most recent data, because I really think it's relevant to the way we think about how bacteria and fungi interact together. This paper, we just published it in September in MBio, where we showed that the bacterium and microbiome interaction underscores microbial dysbiosis in a familiar Crohn's disease. And the good news, it went really, literally viral. I was trending like Trump in Twitter at that time. So just a quick summary about that. We looked at families with CD, and then we looked at their members of the same family with no CD as a control. We also looked at the families who live in the same region but with no CD. And why this is important? I think this is important because sometimes, let's say Crohn's disease, as you know, it has genetic, environmental, and microbiome. So looking within the same family, you at least reduce some of these confounding factors. As I said, I'm just going to give you a summary of this. Here we have the fungal genera correlation. This is in CD patients, and I have Candida here and Saccharomyces, and you can see when the color of the circle is red, it means they antagonize each other. So in CD, they definitely antagonize each other. When you look at healthy people or their relatives, there is no interaction. So this is one of the correlation which I think it's relevant. We also looked at, what we found out in this study is that there are 3 organisms which are really significantly increased in people with CD or patients with CD. The Candida tropicalis, and because Candida tropicalis, like again, is a fungus, we tried to look at what does it correlate with in the bacteria, and you can see it correlated with Escherichia coli as well as with Serratia marcescens, okay? And again, taking the same idea, we want to see how these organisms interact together and influence the host immune response. As you know, in the gut, these organisms don't live floating around, they live inside a biofilm. And lucky for me, because I've been studying biofilms for more than 15 years, and we published a lot in that area. So what we did, we took these organisms and mixed them and tried to see, do they make biofilm? And this is CT alone. And notice most of the organisms are yeast-shaped. When you put it with E. coli, you start to see a little bit more. But when you put it with Serratia marcescens, very high filamentation. And also when you put the 3 together. Now, the other thing we did is we measured what is the thickness of the biofilm when you have the 3 organisms together. And here you are. This is E. coli alone, Serratia alone, Candida alone. And as you go, and this is the 3 together. Very thick biofilm, and this is quantitation for the thickness in microgram, and it is statistically a significant increase or making robust biofilm, okay? Now, this is some microscopy picture, scanning electron microscopy. This is Candida tropicalis and E. coli, and look at this. They love each other. They just are so close. And so when you have the 3 together, again, you see the filamentation, and this is a higher magnification. So this is transmission electron microscopy showing the same thing. Everybody says, who cares about fungi? It is very little of it present. Well, let me show you why you should care. Look at this. This is one fungal cell, and this are all bacteria. It's a huge in size, and they are interacting together very closely. What the difference between them, E. coli, as I showed you, is intimately attached to the fungi, and here you can see very close, whereas when you look at Serratia, what it does, this is better, you can see it has these filaments where it connects to the yeast, but also here to the yeast and also to E. coli. So basically, Serratia is building a bridge between these organisms, and they are all within the matrix very nicely, really forming a basic, strong biofilm. So again, because of the time, I am showing you what I think is happening, and that's why I think you should start paying attention to us fungi. Okay? I think there is cooperative evolutionary strategy that developed to cooperate between fungi and bacteria so that they help each other. Okay? So for fungi, you gain virulence factors. One of the things you can see is there is a lot of filamentation. And when fungi or candida filament, it's able to invade and cause damage to the GI tract. What is the benefit for bacteria? Bacteria, they develop antibacterial tolerance. So there is a study where it showed you have E. coli and candida inside the biofilm. E. coli susceptible to antibiotic. When you put it inside a biofilm, it is tolerant. And then when you use some enzymes to break down the biofilm, guess what happened? It became susceptible again. So this is the two benefits of the term. And unfortunately for the host, this is an inter-kingdom cooperation that impacts the host immune system. I have a lot of data on that, but I'm not going to share it with you now. And under the influence of the enteric pathogens and immunomodulatory components of fungal biofilm, because we showed that there is, like, for example, beta-D-glucan, which is known to produce immune stimulus, levels of pro-inflammatory cytokines increase, causing oxidative damage and apoptotic cell death. And this is how I put it all together. Another paper which just came out in Wound, and basically bacteria and fungi are inside a biofilm. And here, what I'm proposing, when you have this, you have inflammation, increase of all these cytokine and markers of pro-inflammatory inflammation. When you try to use zymolase, which disrupt the microbial biofilm matrix, use antifungal probiotics, you are able to kill these organisms, and guess what? You will have a decrease in the inflammatory process. And this just came out 3 days ago in MBio, where they asked me to write a commentary on it, a cooperative evolutionary strategy between the bacterium and the microbiome. These are the people who I work with from my lab. This is the group from the GI in Case Western, and of course, my dear friend, Michael Laderman, which you all know, as well as Nick van der Berg, and this is the funding. So this is the last slide. This is my son, and as you can see, I call it the wild type. You know, there is resistant and there is wild type. So when he was young, relatively young, he used to do BMX, and this is basically the ceiling, and he's upside down. So I used to tell him, please, every time I come and watch you, I have heart attack. Can you do something more safe? He sky jumped. Thank you very much.